Okay, welcome back to part 4 of the Journal Club series looking at Man and Hansch's paper on metabolism and motility in prebiotic structures. We're going to briefly look at the chemicals used, and then we're going to talk about the chemistry that was happening inside these interesting protocells that were produced. So, substances used. We used a pH indicator, thermolphthalene, which is blue above a reasonably high pH and then tends to yellow or colourless as the pH falls. Use a colorant to dye the nitrobenzene droplets, otherwise it wouldn't be visible. This is red ore, which is an azo dye. It's called an azo dye because of the nitrogen-nitrogen bonds which are linked together aromatic rings. These produce some very nice colours. Very nice colours which are also nicely toxic, and you may remember from a few years ago, especially those of you who like a nice brightly coloured chicken tikka masala, that there was a bit of a problem with contamination of a range of spices and colours used in Indian cooking by this dye, Sudan 1. Again, it's an azo dye. We'll note the nitrogen-nitrogen double bond joining together aromatic systems. So very vivid colours it produces. It's also known to be carcinogenic in animal models and probably carcinogenic in humans. So the Food Standards Agency eventually had to withdraw, I think it was about 400 products in the UK. Here's nitrobenzene, and there's August Kekulé with his fabulous sideboards. You may remember from the lectures, Kekulé was the chap who first figured out the structure of the benzene molecule. Benzene C6H6 was discovered by Michael Faraday in 1925, and no one could figure out how the heck this molecule was put together until Kekulé, after catching the last omnibus home, as the story goes, fell in front of his telly. Wouldn't have had tellies in those days, would they? Fell in front of his fire and dreamt the six carbon atoms forming a ring, a so-called aromatic ring. So nitrobenzene is an aromatic compound. It's oily, it's hydrophobic, which is why when it's dropped into water it coalesces into individual droplets. If you want to know more about aromaticity, and I would recommend you do, the reference on the slide is a useful one. Okay, oleic anhydride. The nitrobenzene droplets that we used contained this molecule. I'm going to come back in a moment and explain a little bit more about its structure and also its name. Another molecule which is important was oleic acid. This is a fatty acid, so it's a long chain of carbon atoms with a carboxylic acid group at one end, often found in olive oil. Just for nomenclature terms and completeness, I'll mention the fact that this is what's known as an omega-9 fatty acid, one of the so-called essential fatty acids. What that means is that we must get them from our diet. We can't make them de novo. We can take them and we can make them a bit longer using enzymes called elongases. They get made a bit shorter in a sequence of steps in the beta oxidation process. We can add and remove double bonds using enzymes called saturases and desaturases, which is good because we use them for a lot of things. But the point is we can't get them from our diet, hence essential. Now this one's said to be an omega-9 one because of the position of the double bond. If you start counting carbon atoms, starting with the carbon atom furthest away from the carboxylic acid group, you'll see the double bond comes at the 9th carbon atom, hence omega-9. Here's another example, one that wasn't used by Hansch, but I thought it included just as another example. This is an omega-6 fatty acid, again one that's essential in our diet. Said to be 6 because if you start counting from the terminal carbon atom furthest away from the carboxylic acid group, the double bond comes at the 6th carbon atom. Okay, now oleic acid is something that's produced during the course of the reactions. I'm showing the synthesis here effectively of oleic anhydride from 2 oleic acid monomers. So you take two oleic acids, you react them together, and you synthesize oleic anhydride. Now it's called anhydride because, like a lot of syntheses, it loses water. If you do the sums, as I've done there, you'll discover we go from a C18 to a C36 with a bit of water left over. Of course, oleic anhydride does have a Sunday name, but I think it's worth using the less proper name here because it does describe not so much its structure but how it was synthesized. The aqueous medium in which this process is taking place contains things called Oleate micelles. It wasn't specified in the paper, but I'm assuming he used sodium oleate, which is the substance given there. You dissolve it in water, and it will dissociate almost immediately to produce the Oleic anion, plus the sodium ion, which will wander off and doesn't take any part in this process, as far as I know. These things are amphiphilic, as we discussed in the last video. It's got a hydrophilic head, the carboxylic acid group, which likes water, and a big long hydrophobic tail, which doesn't like water. So it does things like self-organising, in this case in the micelles. What Hansch and his group did was have micelles containing different pHs, which was the reason why he was able to demonstrate chemotactic movement towards higher and lower pHs. Read the paper if you're not sure about what he did there. Okay, so the experiment. This is effectively the metabolism of our protocell. You get oleic anhydride, it comes in contact with a bit of water and spontaneously hydrolyzes back to 2 oleic acid molecules. These carboxylic acid groups, at the end, as we've noticed, and when we put them in water, of course, they partially dissociate because it's a weak acid, produce again oleic anion. I haven't drawn the whole thing here for room, plus some protons, which are going to change the local pH and do so quite significantly, as we'll see in a bit. We get movement. We get the little red droplets, which contains nitrobenzene and oleic anhydride, which is metabolizing to oleic acid, changes the pH and moves around. Eventually the movement stops, basically because all of the oleic anhydride has been hydrolyzed. So here's a little schematic of what's happening. The red circle is the nitrobenzene droplet surrounded by water, and there's a single Oleate, oleic anhydride molecule. It comes into contact with the boundary between the nitrobenzene and the water and immediately hydrolyzes to produce, as we've seen, 2 oleic acids. These dissociate themselves, pumping protons out of the system, so we're changing the pH around the droplet. It's quite a big change. 4 units change in pH is significant. If you remember, pH is a log scale, so that's a 10,000 times change in the concentration of hydrogen ions. We also see a change in something called the interfacial tension, which is effectively a surface tension between 2 liquids. Quite a significant change, which produces one of the Marangoni instabilities we talked about, I think, in the first video. And we get movement. So we get our protocell metabolizing and whizzing around its little environment and producing small changes in the environment in terms of the pH. Okay, I'm nearly finished now. I hope you've enjoyed this series. I will certainly be back later on with guinea pigs, humans and carrots. But just to sum up a few things. What Hansch isn't saying is this, that how protocells evolved on Earth. What he's doing is modelling a simple system which self-organises, has a metabolism, which produces motility and an environmental change. There's lots of groups around the world working on these sort of things. And if you're interested, I've given you some references in previous videos, and also have a read yourself. Plenty of interesting information out there. Here's something to think about. This is going back to Oparin's definition of life. How many of these things are actually happening in the system we have here? I'll give you a clue. I don't think it's many, but have a little wander through and decide what you think is happening here. Okay, that's it. Thanks very much for listening, and as I mentioned, I'll be back reasonably soon with the first part of guinea pigs, humans and carrots.